Really quickly tonight, because it's all the way in December and I don't even know how we got here, but here we are. Happy holidays, everyone. Thanks to everyone for actually coming out in December. That's miraculous. It's just sort of how our schedule went. We usually take a hiatus this month, but I'm really glad that people came. We have a really interesting lecture tonight. Well, actually, let me say maybe a couple of words about the Stem Cell Research Center, which I usually try to do, but I don't always manage. Some exciting goings on to highlight a couple of things. We actually, for the GMP facility, Good Manufacturing Practice facility, many of you have heard me talk about in the past, we are fully operational as of this month, really running our first manufacturing product starting in January. So super exciting for all of us that have been working hard on that. We are waiting with bated breath to hear about the Translational 1 grant that Anand submitted today. Everybody say yay. And that might take a while, but in the meantime, we're waiting to hear from CIRM about our Shared Research Lab grant, which will support our core facilities for use by lots of investigators from across the campus. So we hope that that's going to go well. We thought we might hear in December, but it was a huge effort to get it in. So a particular shout out to Craig Walsh and Matt Blurton-Jones and Matt Inlay and Randy Berg from the center, who really made that happen, and a whole team of people that were behind them in terms of the grant submission. With that, a shout out to Brian Cummings and Randy and Abby Enriquez from the center, who have really kept this community lecture series running. As you know, we're trying to build our audience back up to kind of where we were pre-pandemic. So tell your friends and neighbors that it's so much more fun to be here in person. We're working on that. With that, we have a great lecture tonight, and you have not spoken before, from Anand Ganesan and his clinical scholar, Catherine Hinchee Rodriguez. And this is really kind of a unique opportunity. So as you know, we try and balance these lectures, either having a basic scientist and a clinician, or more recently, we've been working with having trainees and their PIs speak at the same time. Tonight, we're going to have a different treat, which I don't think we've had so far. So from two people who really work at the, not just translational science, but actually implementing that from their labs and into patients, and how those two things are really connected. So hopefully ask them a lot of questions and that will come out. Do you want to talk about the P30 and the skin center a little bit? Or, yeah, then I'm going to let Anand do that.
All right, well, thank you everybody for being here tonight. I know it's rather chilly Friday, or I mean rather chilly evening, Tuesday evening in December. See, I forget my days. As Dr. Anderson mentioned, one of the things that the Stem Cell Center does, which is fantastic, is they support the Skin Biology Research Center, which is a great center that promotes interdisciplinary skin science research that ranges from the clinic all the way to basic and back. And so that's a great aspect of the Stem Cell Center and the Stem Cell Research Center in general. And one of the other things that the Stem Cell Center does, which is actually quite unique, is they're one of the few centers on campus that actually has slots on their CIRM training grant for clinical fellows or for physician scientists. So if you don't know, physician scientists are individuals who do science and they see patients. It turns out if you survey all of the physicians in the United States, only 1% of them say that research is a significant part of their professional activity. That means that 99% aren't doing any research. If you look at that 1% who say that, if you look at the amount that they're doing research, it's actually minimal overall. And the numbers that are NIH funded are probably 1/10 of the percentage of the 1%. So it's a very, very rare field. And why is that? Well, there's a couple reasons. One is because it's very difficult to find training opportunities for physician scientists. It takes a long time to train. You go through many stages of your training, and you have to learn how to integrate seeing patients and also doing research, which is a big challenge. So today, we're going to give you a nice example of what you learn when you're doing translational research from the bench to the bedside. So Katie completed her undergraduate at Duke University and an MD PhD at UT San Antonio. And then, we met a couple of years ago, and she said, hey, I really want to devote myself to doing research and developing my research career. And so fortunately enough, she was able to apply for the CERM training grant and get a slot. And in the last year and a half now, we've been working on really trying to understand melanocyte regeneration, which is kind of really interesting because for a long time, everyone thought the melanocytes you have in your skin are there always, right? The adult melanocytes are what you have from what you're born with, and that's what it is. But lo and behold, if you look at disease, and vitiligo is a great example of that, when you lose your melanocytes, they can regenerate. And so where do they come from? How does that happen? And how can you promote that to treat disease? So Katie's going to tell you a little bit about that today. So with that. You have her own mic. Perfect. Okay. Yeah.
Okay. Hi, I'm Katie Hinchee Rodriguez. Thank you for the nice introduction, Dr. Ganesan. I am a CERM clinical fellow, and the talk tonight is Novel Diagnostics and Treatments for Vitiligo, a story of melanocyte regeneration. So just an outline of the talk. So we'll just be doing some basic skin and melanocyte biology. We'll go into vitiligo, how it develops, but also how it affects the people who have it, as well as what are the treatments right now and trials right now for vitiligo. We'll go into Resell, which is a specific clinical trial, and we'll go also into imaging and its a relationship with understanding vitiligo, as well as understanding the unique role of keratinocytes in vitiligo. So in our skin, it's made up of, oh, let me turn off the, yeah. Let's turn it to dim. There we go, better. So our skin is made up of three main layers. We've got the epidermis, which has the keratinocytes. These are the cells that are kind of like the protective layer of cells in the epidermis. And then there's the pigment-producing cells in the epidermis, which are our melanocytes, and they're at the base of the epidermis. And then we go to the dermal epidermal junction, the DEJ. So that's where fingers of the dermis project into the epidermis and kind of create that like poking into it. And then you go into the dermis after that. That's where the fibroblasts are, and they make the collagen and elastin that act as the architecture and support for our skin. And then underneath that is a layer of subcutaneous fat. Not as interesting for me today. So for melanocytes specifically, they synthesize melanin. Melanin is the pigment that gives our skin color. It's packaged into little organelles called melanosomes. And melanosomes are transported from the long spindly arms of the melanocytes to nearby keratinocytes. And then the melanin is then kind of created into a little hat that the keratinocytes wear that protects them from UV radiation. All skin tones really have a similar number of melanocytes. What really is more different is kind of the number of melanosomes and how those melanosomes are packaged. So darker skin tones will have more melanosomes that are differently packaged compared to lighter skin tones. And melanocytes are activated by UV radiation to produce more melanin, which is that UV protective pigment. So Dr. Ganesan kind of mentioned that it used to be thought that there wasn't really any regeneration capability for melanocytes, but there is. So what we know right now is that the hair follicle is one of the main sources for melanocyte stem cells. So within the hair follicle, so this is our hair follicle here, as you can see, it goes through the epidermis, and then a lot of the body is within the dermis itself. The melanocyte stem cells live in a little pouch on it called the bulge. And so some melanocytes do live at the bottom of the hair follicle, that's why hair has color. And then the stem cells will also crawl up the hair follicle, and they will populate as mature melanocytes along the bottom of the epidermis. Another source of melanocyte stem cells in our skin is actually like within the epidermis itself, so epidermal stem cells. And so these are kind of like populations that are just intraepidermal. we have melanocyte progenitor cells, which will then mature into, mature melanin-producing melanocytes. But what happens when melanocytes are lost? So what happens is that people, if they're losing their melanocytes in a certain part of their skin is going to lose all pigment in that area. It's going to turn white. And so this is actually from a really fabulous photo shoot from Imperfectly Perfect, which is a vitiligo group in Wisconsin. And they did a really nice photo shoot of all these different people who came in to kind of show and represent themselves as individuals with vitiligo. So vitiligo is when the pigment is lost in skin. This is when the melanocytes are gone. So vitiligo, how does it happen? So there's kind of like three ways we can think about it. A lot of times we think of the inflammatory autoimmune aspect of it, which is where our body's own immune cells, like the T cells, travel in through blood vessels that are just already in the dermis, and then they're drawn into the dermis. The T cells are drawn in, and they kind of produce factors that affect our keratinocytes, and the keratinocytes then produce factors that damage our melanocytes, or the T cells directly, by producing antibodies, more directly affect and kind of destroy our melanocytes. But in addition to the role of autoimmunity, there's also like an inflammatory component that's related to our keratinocytes and our fibroblasts. So we also know from other data that keratinocytes themselves, without T cell roles, keratinocytes can produce factors that can make the microenvironment for the melanocyte inhospitable. And fibroblasts can also do that as well, producing factors that are going to affect melanocytes to produce then their own factors that make the melanocyte, or keratinocytes produce factors that affect the melanocytes, and just making the environment inhospitable to them, so that we're not having melanocytes surviving and we're not having melanocytes regenerating. So Dr. Ganesan mentioned that vitiligo can be a great melanocyte stem cell regeneration model. And it's very striking visually when you see that regeneration happening. So this is images of an individual with vitiligo. And so this is them repigmenting. So remember, the melanocyte stem cells, a lot of it is in the hair follicle. So as you can see, there's little hairs here. And so a lot of times when people repigment in vitiligo, you're going to see the repigmentation happening in this very particular pattern because it's happening around the hair follicle. And so this is kind of like patients with vitiligo kind of talking about what their experience is like with this condition.
I'm Indian with white patches.
I'm an African American with white patches.
I am Hispanic with white patches.
I am black with white patches.
I'm Caucasian with white patches.
I am Asian with white patches.
I'm human with white patches.
They've heard it all.
It looks like your skin is coming off.
You look like a cow.
I'm burnt.
I'm ugly.
You're an alien.
So we heard these individuals talk about kind of, some of a lot of them are proud to be individuals with vitiligo, but they do sometimes suffer negative consequences. So for vitiligo quality of life, there's kind of like, we can think of two areas, like the features of vitiligo, how the disease itself or the condition itself affects people, and then also kind of more social and religious aspects that can affect individuals with vitiligo. So vitiligo, sometimes it's asymptomatic, but sometimes people actually have itching and burning that can be uncomfortable, especially if it's active disease where they're actively depigmenting. And then also what they can kind of feel, there's an unpredictability. Is it going to depigment further? Am I going to repigment some more? And so that kind of is distressing in and of itself. And also a sense of imperfection or being different from others. And then as well, they will feel sometimes that they don't have a control anymore over their body. They don't have as much body integrity as they had perceived themselves previously having. So what is available for vitiligo patients that are seeking repigmentation? So right now on the market, there's kind of three areas we can think of: immunosuppressives, melanocyte activating, and melanocyte replacement and regeneration. So within immunosuppressives, and a lot of the immunosuppressives have actually been used for a very long time. So there's topical steroids, topical tacrolimus and plecrolimus. The newer one is the topical JAK inhibitors, such as Opzelura. And we'll talk a little bit more about JAK inhibitors soon. And then there's oral steroids, as well as phototherapy. Phototherapy is when individuals get UV light that's controlled. It's just the UVB, not UVA. So it's actually quite safe, and it stimulates the melanocytes to produce pigment, but it also has an immunosuppressive effect. And so in terms of melanocyte activating, we've got the phototherapy. And then what if we want to replace our melanocytes, then we need to use grafting. So the grafting techniques currently are punch biopsy grafting, where you take a plug of skin out from unaffected skin and then put it into a punch biopsy hole that is in the vitiligo skin. So you've got these punches of normal skin that are going to repigment out. As well as suction blister grafting, where suction is actually used to create a blister in the epidermis, and we use that epidermis as a graft. And then there's melanocyte keratinocyte transplant procedure, or MKTP. And then there's Resell grafting. Resell grafting is a kind of like a further development of MKTP, where you're taking this a sample of skin that's unaffected by vitiligo on individuals. You're dissociating all the cells in that sample, and then you're putting it back onto their own vitiligo skin. And that kind of like changes the microenvironment to promote the graft taking. In terms of clinical trials that are currently being done here at UCI, we have oral JAK inhibitors. So the oral JAK inhibitors are going to break that autoreactive T cell inflammation cycle. And for the trials that are upcoming within that, it's INSIGHT phase 3, for povorcitinib, AbbVie phase 3 trial, for upadacitinib, or Rinvoq. You might have heard Rinvoq on the TV for eczema. There's also a currently enrolling study where it's an injectable anti-IL-15 antibody, and IL-15 is part of that pro-inflammatory cycle. So it's also breaking that T cell pro-inflammatory cycle. And that's a trial between Amgen and the National Institute of Allergy and Infectious Disease, NIAID, which is a phase 2. And then our closed trial with Avita Resell, so that's skin grafting, where we're replacing the old skin microenvironment with new healthy keratinocytes, melanocyte stem cells, and fibroblasts. So we're replacing everything that has some kind of effect on pigment regeneration and kind of putting in stuff that will actually promote it now. So the JAK inhibitors that we mentioned, the topicals and the orals, so JAK inhibitors are involved also in changing the melanocyte microenvironment. So the way they work, kind of like overall, JAK is part of a signaling pathway. It's a protein. And what it does is promote the transcription of genes that increase inflammation and autoimmunity. If you have a JAK inhibitor, it's stopping JAK from causing that promotion of inflammation and autoimmunity gene expression. And so the JAK inhibitors they will work on the aspect that's with T cells, the aspect of keratinocytes, the aspect with fibroblasts, because all of those have that inflammatory cycle component to them. And so then with the JAK inhibitor, you often see, like, again, that repigmenting pattern, because the melanocyte stem cells are able to actually survive and come up from the hair follicle and create new melanocytes that will cause that kind of hair follicle follicular repigmentation pattern. But what if there are no melanocyte stem cells left? So when we have JAK and the JAK inhibitor works, we are saying, okay, there's still stem cells in the hair follicle. We can still get something to come out of there. But what if there's no stem cells left for that even to work on? So that's where the Avita Resell skin grafting comes in. So Avita is the company that developed the technology Resell. Resell is this device that we're seeing right here. So with the Resell grafting, you obtain a normal skin from a donor site, and you basically use the device to prepare a cell suspension of epidermal cells, but we also believe there's fibroblasts. We know that there's fibroblasts as well, not just the melanocytes and keratinocytes, but also fibroblasts from the epidermis are getting put into the cell suspension. We use a laser And so that's kind of like a representation of the cells that we're putting back in. And we use a laser to kind of take the top layer off of the vitiligo skin and prepare it to receive the new healthy mix of skin. And then we just put dressings on to keep everything in place for the first week. So here's the laser blade. So this is an individual who has chronic vitiligo. It's not repigmenting. We will take a laser, we will ablate through the epidermis and then slightly into the dermis, just a little bit. And then we will apply all those new healthy cells, the keratinocytes, melanocytes, fibroblasts, and the melanocyte stem cells. So we're replacing the entire microenvironment, essentially, in the vitiligo areas that are being treated. And then what we get then is a healthy repigmenting skin post-grafting. We've got our hair follicle, we've got our stem cells in the bulge, we've got stem cells traveling up and actually being able to survive now in the epidermis. And instead of the keratinocytes and fibroblasts producing factors that are negatively impacting the survival of melanocytes, they're actually producing factors that allow the melanocytes to thrive. And in terms of data that's directly from the phase 3 trial, so what we're looking at here is how many people received at least 80% repigmentation at 6 weeks, 6 months, week 24. And I just want to note in this trial, everyone did 3 days of phototherapy a week throughout the duration of this trial, which was a total of a year. So at just halfway point, 6 months, 36% of the individuals treated had gotten at least 80% repigmentation. And then in terms of how many people received at least 50% repigmentation at the halfway point was 56%. And then for how people felt about their repigmentation, 60% of subjects said that they felt their Resell treated vitiligo was much or very much improved at the 6-month point. And then kind of visually, these are some examples of results that we're seeing with Resell. So this is an individual whose vitiligo has looked this way for 4 years. It's been stable. Something's keeping it from repigmenting. So this is the area that was control, not treated, and this is the area that was treated. And what we see is the repigmentation, and obviously this is an example, but really fantastic repigmentation at the 6-month mark. In this individual, they had had their vitiligo being stable and not repigmenting for whatever reason for 10 years. This was the control area. This was the area treated with Resell. And at 6 months, they too also had a pretty good amount of repigmentation in the treated area. But kind of the question is, well, who is going to be more responsive? So we've got some really great responses, but not everybody was an 80% repigmentation individual. So why are some people responding more than others? And why do some areas not repigment still with Resell? So we wanted to use non-invasive imaging to identify who will or won't respond to treatment. So kind of going very briefly into non-invasive imaging modalities, you can kind of envision them as optical biopsies. So these are 2 examples, Vivisight, Viviscope, but for us, we're interested in MPM, multiphoton microscopy imaging. And the commercial device that does this is called MPT Flex. And the group that we collaborate with actually created their own device, and we'll go into that later. So MPM, multiphoton microscopy, has a lot of strengths. It has extremely high resolution, which means not only do we see cells, we see what's inside the cells. We can use color that will allow for contrast between different parts of the tissue. And because we can see within the cells, we can actually see metabolic changes within individual cells. And so right here, this is an example of melanocytic nevus, or just a pigmented mole. And so we've got our epidermis, our dermis, and we're kind of looking at it from top. This is the MPM imaging, and we're kind of going through it in slices, transverse slices going from top to bottom, from epidermis down into dermis. And so we're seeing green is the keratin and the keratinocytes. And as we go down, we start to see blue, which is the collagen, which is the matrix and architecture that's within the dermis. So we're going down from epidermis to dermis. So the Baloo group, who we collaborate with, actually developed their own MPM imaging device, which is called FLAME, Fast Large Area Multiphoton Exoscope. Here it is. They're developing a new one that's actually mobile, so you can move it all over the place, which is fantastic. And again, what we're seeing here is, epidermis, dermis, and then when we look at it through MPM, we see the keratin, the green keratin of the keratinocytes, the red melanin and the melanocytes, and then also like within the keratinocytes themselves as they wear those little hats, and then the blue collagen of the dermis. And I kind of just want to orient you into how we see the dermis within MPM with FLAME. So we're looking at top to bottom, epidermis to dermis. And remember, at the dermal epidermal junction, we have the dermal papillae, the fingers that poke up into the epidermis. So if we took an epidermal papillae and we sliced it in half, it'd be like, slicing an icicle in half. You're going to look at it from the top to bottom as kind of like a circle. And so when we look at an individual with vitiligo, but we look at their unaffected skin, so the normal skin, with FLAME, we see kind of this pattern of dermal papillae in the blue, surrounded like a very organized collagen within the dermal papillae, surrounded by melanocytes and melanin in a very organized manner in the red. And then we're seeing some of the green, the keratin of the epidermis, kind of coming in through like just natural skin folds that happened. And then this is individuals who received cell suspension grafting in their vitiligo areas. And this is what their FLAME, their MPM imaging looked like at 1 month post-procedure, so post-replacement of the entire microenvironment. So we have patients 1 and 2. You can see the collagen is very disorganized. The melanocytes are intermittent, kind of also disorganized. Whereas in patient 3, we were seeing more of an organized collagen and dermal papillae with that organization of the melanocytes and melanin around the dermal papillae, similar to how we saw it in unaffected skin. So it's interesting to us then to kind of, where the study is developing, we want to follow these individuals and see how they repigment. And because, are these people less likely to be responders than someone who's already seeming like they're reorganizing their environments more similar to unaffected skin? So then the question becomes, okay, so what are the local factors then in melanocyte environment that are responsible for inhibiting or promoting repigmentation? So in chronic or stable vitiligo, these are individuals that their vitiligo is not progressing, but it's not improving. It's just been the same for years. So this was an individual, remember this from Resell, but they repigmented in a lot of it, but this area right here didn't repigment. So why did this area repigment and this immediately adjacent, also treated area not repigment? So one of the techniques that the lab has used, the Ganesan lab has used, is called single-cell RNA sequencing. So that will allow you to look at individual skin cells' gene expression. And you can use this to kind of explore that question that we have. So you can take your skin sample either as a suction blister, where we're using suction to kind of lift up and take just the epidermis as a tissue sample, or you can do a punch biopsy, where you kind of like cut in and you get a plug that has both epidermis and dermis to it. You can then use this like skin sample tissue to then do a single cell analysis. So you're looking at each individual cell within that tissue sample that you got. And then based on the gene expression data, you're looking at, okay, what are our different populations of cells as defined by unique gene expression signatures? And then you can organize them into little populations of cells that you have within your skin sample. So using this technology, the single-cell RNA sequencing technology, the lab, what they were investigating is there a unique population of keratinocytes that could be affecting the ability to repigment or not repigment? And so what they found is that there is a unique stress keratinocyte population in stable vitiligo. So when they looked at the different types of cell types in their lesional, so vitiligo, or non-lesional, so unaffected skin, they found that there was a very specific population called stress keratinocytes that was only in the vitiligo skin. And it's kind of, called a stress keratinocyte because when you look at the gene expression that this population has, this only vitiligo population has, a lot of it is pro-inflammatory factors, such as CXCL10, CXCL9. There's some specific keratins and also other pro-inflammation factors like S100A9, S100A8. So we're seeing that this expression is really only in the stress keratinocytes. So another technique they use to kind of investigate this unique stress keratinocyte population is called mitochondrial clustering. So I mentioned with MPM, you can look at metabolic differences. And so this is one of the ways. So with mitochondrial clustering, you're able to quantify mitochondrial organization. So mitochondrial clustering, it's represented as beta value. So as you have increasing beta values, you have increasing mitochondrial fragmentation. So mitochondria fuse in a state of metabolic called oxidative phosphorylation or OXPHOS, and they stay separated or fragmented when they're undergoing glycolysis. So if you have increasing beta values, you're having increased mitochondrial fragmentation, and that means increased glycolysis rather than oxidative phosphorylation. And what you see in normal skin is that it's a depth-dependent variation in the metabolism. So the bottom of the epidermis is more glycolytic than the upper dermis. And so with non-lesional skin, it's depth-dependent. So let's take a look here. So this is the non-lesional, the normal skin, and we're seeing this very distinct pattern. Right here, we're going deeper into the epidermis. So we're starting off at the top of the epidermis, more like oxidative phosphorylation. And then as we go deeper into the epidermis, we're going higher, which means we're having more glycolysis instead. There's a switch to glycolysis in this normal skin. Whereas when you look at the vitiligo skin, you don't see this pattern. It just kind of flatlines and stays at oxidative phosphorylation. So you have this kind of unique signature here. Now, when you look at the stress keratinocyte population, they also have this, this very interesting response. So if you're looking at the differentially expressed genes between stress keratinocytes versus just other keratinocytes, you see that there's a way increase in oxidative phosphorylation genes in the stress keratinocytes compared to other keratinocytes, and not so much glycolysis. Now, and this is an individual with vitiligo who got punch grafting. And so they're a responder. You can see there's like increase in pigmentation where the grafts were put in. So in this responder, at baseline before the grafting occurred, we see kind of that flat line pattern, just OXPHOS. And then after 10 weeks, as they're repigmenting, we're seeing that change. We're getting that non-lesional skin signature where we were going from OXPHOS up to glycolysis. And then in an individual who also got punch grafting, but's a non-responder at 10 weeks, again, they're just flatlining. At 10 weeks, they're still flatlining. And so from further data from our lab, and this was actually just came out in GID this year, what they wanted to do is also look at MPM specifically and correlate that to the metabolic changes. So through MPM, they were able to see the melanocyte migration. So this is acceptor or vitiligo skin. This is donor, normal skin, and you can see all those little dots. Those are melanocyte caps or melanin caps on top of the keratinocytes. And so in this skin where grafting happened, the blue arrow is the graft spot, and the red arrow is just an adjacent site of normal skin, of vitiligo skin. And so in the graft site, I know it's hard to appreciate with this picture, but you kind of see these little squigglies. These are the melanocytes coming back into the graft site. And we see in the adjacent site with the hair follicle, and remember the hair follicle is where we can get some of our melanocyte stem cells, we're seeing an increase of melanocytes coming into the area. So coming from the hair follicle, potentially also migrating into the area. And then this was correlated to the metabolic signature that we've discussed before, where if you're a responder, you're getting kind of this return to the non-lesional metabolic signature compared to a non-responder. So in summary, for kind of that part of the story, changes in keratinocyte expressed factors contribute to stable vitiligo. So you've got your stressed keratinocytes, they're expressing factors that create a local inflammatory loop and promote melanocyte destruction and decrease melanocyte migration in chronic vitiligo. So our next big question was, what is the impact of stable vitiligo keratinocyte melanocyte signaling on melanocyte maintenance and repopulation? Well, first we need to look into what factors are responsible for resistance versus response. So AIM-1, so this is for the CERM clinical fellow position that I have. This is part of what I wrote. AIM-1, we wanted to use the intrinsic spatial heterogeneity in partially therapy responsive vitiligo to identify the factors that promote melanocyte maintenance and regeneration. So to do this, we want to take punch biopsies. Oh, it didn't show up as well as I wanted. We take punch biopsies and we take them from unaffected skin, repigmenting skin, so this is individuals undergoing treatment, and non-repigmenting on non-responsive vitiligo skin. And we want to compare all of these to each other. And to do this, we're going to use spatial transcriptomics. And just very briefly, with spatial transcriptomics, you take your punch biopsy, and what you're able to do is kind of using all that similar RNA sequencing concept, you're able to look at like individual cells and their gene expression profiles, but you're also then able to map that gene expression profile onto the true space, the true like spatial orientation and relationship between all these cells within the tissue. So you can see the communities, the environment, and how these cells are interacting with each other, and it elucidates a lot more information. And then we also want to build a model to explore the relationship between different factors involved in melanocyte regeneration in vitiligo. So we can find out more about the factors with spatial transcriptomics, but we also want to then create a model where we can implement what we've learned and kind of further understand, well, how are the different ratios of melanocytes to keratinocytes and different ratios of these factors actually affecting repigmentation. So that goes to AIM-2, where we want to establish a patient-derived 3D skin culture model to test that changes in cell-cell interactions influence the melanocyte repopulation in vitiligo. So we get skin discards from surgery, we isolate them, I culture them, and I'm isolating melanocytes, keratinocytes, and fibroblasts. So I get a chunk of skin, I do cell culture and I'm trying to get these three populations. I'll use flow to identify specifically that it's these populations. So flow cytometry is a technique where, again, you're looking at individual cells and you're looking to see, do they have a marker that tells me it's part of this cell population? So that way you can feel confident in the identity of the cells that you're culturing, because these are all from patients. This is not something you buy from a company. And then we want to apply these cells onto an engineered dermis to create the patient-derived 3D skin model. And this is just an example of some of the flow cytometry data. So we have the patient-derived cells, and we're looking to see through the flow cytometry as they go individually through this little channel, do they have the marker for the population that we're interested in? So for us, I'm interested in melanocytes and keratinocytes. And so this is just an example of, we're looking at melanocyte marker. So increasing means we've got more melanocyte marker. And we see that there's this population of cells that are kind of positive for the melanocyte marker. And then with keratinocytes, we're looking at a keratinocyte marker and we're finding a population that's positive for our keratinocyte marker, so we can have a better idea of how much we're isolating and if they're actually there. And these are just images that get taken as they go single cell through that channel. And these are just specifically the ones that are melanocytes here, and then the ones that were identified as keratinocytes. So in summary, in stable vitiligo, so even though it's not considered active, there's still things happening. There's things happening that are preventing the repigmentation from happening. You've got T cells affecting the keratinocytes, which then inhibit the melanocytes. You've got T cells directly negatively interacting with the melanocytes, and fibroblasts expressing factors that also affect keratinocytes to make it a hostile environment for our melanocytes. But with repigmentation, you can get it from the stem cells which are in the hair follicle, or they can be in the epidermis itself. If you replace, like we talked about with Resell, you can replace the entire microenvironment and you can get repigmentation happening. You get the melanocyte stem cells are able to be stimulated, they can repopulate and be sustained. And then once we kind of like understand better what factors are involved in promoting or inhibiting repigmentation, we'll better understand the maintenance of melanocytes and their regeneration. So, once we've got melanocytes migrating into the tissue, there's no more inhibition of migration, and in fact, there's promoting factors being expressed by keratinocytes and fibroblasts that are actually going to sustain the melanocytes and prevent that inflammatory cycle from happening. So I just want to thank all of the members of the Ganesan Lab, the UCI Derm Clinical Trials team, the members of the Baloo Lab, Avita, and funding for all the work that was seen here came from CERM or the National Institute of Allergy and Infectious Disease, NIAID, or from Avita. Thank you.
You stole the microphone. There you go. Okay. Don't turn that off, because I'm going to put you on the spot. So first question, we heard a little bit from your mentor about how unusual it is to end up as a physician scientist, right, and really go down this research path. So why are you doing this? Like, how did you come here, and why are you doing this?
There's a lot of factors. There's like Dr. Ganesan said, it's a long career path, so a lot of things are affecting you along the way. I was interested in biochemistry as an undergrad, and I realized that I wanted to understand more of the human impact that my research would have, rather than just kind of tinkering away without seeing anything real happening. So that's how I decided to apply to an MD PhD program. And then MD PhD programs can be difficult. Some people end up dropping out. But I kind of remained interested in research the whole way through grad school and medical school. And I became interested in dermatology specifically. And so when I was looking for research fellowships in dermatology, I actually found there was like an old posting for Dr. Ganesan that was like no longer active. But he actually read my CV and was like, maybe actually you're a good fit. And it's been a fantastic fit because I've been able to have access to basic science, translational science, and clinical research, which for me is clinical trials is not something I've done before. But I've been really impressed, especially with the in vitro trial, with kind of like the impact that they have on patients.
So that was a great answer. Thank you for that. It occurred to me as you were talking, though, because you went the MD PhD route, and there are other folks, like our present company, who don't necessarily go that path, right? And you still end up on a research track. And so maybe Anand, can you tell, like, how did you end up doing research?
Well, I did an MD PhD too, so.
He's a double doctor, and I'm so called out.
Oh, no, it's okay. But there are people who do, actually, it's interesting that you asked that.
I have some slide where I acknowledge you, and it's only MD.
No, it's okay. No, it's fine. But I don't believe in labels, so we're all good. But there are people who do, and it all depends on when you catch the bug, actually, is what it comes down to. Because there are people in residency. We have one actual fellow who's working with Dr. Dai who only caught the bug to do research after she started her residency. We have some who go through the MD PhD route. It turns out actually that the MD PhD route, the ones that actually move on all the way through, is actually lower than you think. In fact, the rate that actually move on are more when they do research during residency than actually during MD PhD. And it's only because of the gap in the time that's the problem, because your life happens in the middle, and most people get drawn away from one reason or another. So yeah.
So I'll cover my embarrassment totally. So a parallel question to that would be and then I'll let you guys ask questions. Dr. Hinchee, Rodriguez. A parallel question to that, which I think is hard to understand for folks who don't work at a university, right?
Right.
Is what are the benefits of an academic medical center, right? And how do you manage that in terms of your clinical work, right? Because release time is a real challenge.
Right.
Yeah.
Right. So
explain release time.
Yeah.
yeah. So this is a great question.
And
So
you should not listen now.
no. She knows this whole deal, so we've talked about it. But a lot of it has to do with the way that everything works. Unfortunately, in research, is that you sort of have to produce before you get paid to produce. So that's always the problem. And in the beginning, you just need people to give you time to do this. And so one of the things that we do as an academic medical center is we make bets on who we give that time to. And a little bit has to do with planning and thinking about what you want to do, thinking about what career direction you want to go in. And then it's just mainly finding mentors that can help you so that we have a good sense that you're making the right bet in that direction. And that's really what it comes down to. And academic medical centers, as you were alluding to, so physicians, why are only 1% of physicians doing research? Well, research doesn't necessarily, I think if you talk to a lot of doctors, they'd be like, oh, I'd love to do, all my clinical colleagues I talk to, oh, I would love to do more research. You're so lucky you get to do research. But the reality is you have to work 3 times as hard to do research because you have to do research to get money to do research, and then you can start doing it, and then you have to work on getting money next time. So it is a lot of work to do that, and then it's not like seeing patients where you go see them and then the money churns right away. It's a lot different. But it makes a much more profound impact on people.
Yes, and why is that?
Well, it's because you hear their problems, you hear what they come to you. So how did this whole project start, if I can just give you this idea, is that I saw some patients and I was thinking, and I was doing this punch grafting procedure, and this is the stem cell question. I started thinking, why are these things only migrating so far and stopping? And why is it that it stops differently in different people? It's not that so much they go away. See, if they went away, everybody would say it's the immune system, and that's what's doing it. What was happening is that they were migrating a certain area and then they were stopping. So that has to be the local tissue doing something. So how is the tissue controlling the stem cell? And we know that happens, right? When you look at your skin, it's not growing infinitely and sloughing off your body to the floor, right? It knows where to stop. It knows how the cells interact with each other, and it knows how far to go. So that was the initial interaction. And then, patients, when you talk to them, we spend all this time we talked about, everybody's interested in vitiligo immunology, which I will not argue that the immune system is important in initiating the disease. But why it doesn't repigment is a stem cell problem. And there are very few people that study that, actually. So, yeah.
Yeah. Perfect. Thank you.
Hi, so you talked a bit in your presentation about NPM imaging. I was just wondering if there are any other applications of the imaging technique.
Yeah, so I think, go back, but so I talked about FLAME. So that's the scope that was developed here by the Ballou team. And it's not just the multiphoton microscopy that FLAME can do, but you can also combine the NPM with FLEM. And FLEM, I can't remember off the top of my head what FLEM stands for, but you're basically using the endogenous factors within cells for like an autofluorescence depending on the energy of the wavelength of light that you're shooting at that cell. And that can give you really great ideas about, not just like we talked about mitochondrial clustering, but that can also give you ideas about metabolism. And when you combine the FLEM together with the NPM, it's extremely powerful because you get all this other stuff that you can use, like signatures that you can use to identify specific cell types and specific states of these different cell types. Yeah.
I can make a comment to that too. You know how like Google facial recognition works? You go and you put your facial image in there and then it takes it into pixels and in individual pieces and then puts together what identity you are from the whole picture. So what NPM can do, what you can start to do is you can start to do feature selection on different cell types. So phlim is actually the lifetime of the fluorescence signal. So not only does each fluorescence have its own distinct wavelength, so you excite it and then the wavelength, the time that each wavelength lasts before it decays differs depending on the substance. So in a way, you can actually figure out what the different factors are that you're looking at, the molecular contrast, by looking at the decay time. So you have the decay time, you have the location, you have the signal, and all of those things when you add together. Do you want to go back to the graph picture, the Resell? So this is one of the things that we're doing. In fact, we have the MPM after the Resell, that one, or after the MKTP, whatever it was that we did. Yeah, so go back. So you see all of these cells here that you're seeing. So some of the red cells are the melanin-containing cells, but what are the yellow cells? What are the white cells? Those are all different cell types based on their own fluorescent signature. And so one of the things that we're doing, and we have funding to do and we're pursuing now, is actually trying to identify what each signal is and what cell type it is. Because it turns out that we can see a certain signal with T cells, we can see a certain signal with macrophages, we can see a melanocyte signature, which is really easy because they have melanin. But you can identify different cells based on what their signature is. So one of the projects we're doing is we're actually looking at wound healing and seeing which signatures come in which cells and at what time, and then how do we identify them. Because then you could do things like, so the big holy grail here is, you see on TV, these dermatology drugs for psoriasis, for vitiligo, for everything like that, which are like $40,000, $50,000. Or you see immunotherapy drugs, which are $100,000, $200,000. You don't know if they're working, and when they work or when they don't is what happens to immune cells. Do they go away? Do they come in? And so now we're using this to actually do that, to try to see non-invasively. And that's a huge thing because not only in pharmaceuticals is the issue seeing it, but it's where to see it. Because most of the methods we have now is you take a piece of skin from here. Is that the right piece of skin? Is that really telling you what's going on? Or is that prone to sampling error? So.
Hi, Katie. That was a beautiful talk. So I have a question about the mechanism of recells. Does it work because the dissociated cells from the normal tissue integrate on the treated site and form a new epidermis, or is it some kind of signaling molecules and growth factors from the normal tissue that's promoting the repigmentation on the treated site?
I think it's a little of both, but I think it's primarily the first. Let me see if I can remember where my Resell slide was. But yeah, so I think you've got we're ablating the epidermis, we're taking away just a little bit of the dermis, but we've got that architecture still there. It's kind of like a 3D, I talked about a 3D skin model where you have an engineered dermis. It's kind of like that. So we've got the dermis, the endogenous dermis there, and we're putting in all the new cells that you would need to create an epidermis. And so it's a new population of healthy cells that have normal signaling and normal relationships with each other. So I do think there is a component of factors that are being added in, but I think it's mostly just kind of the identity and the state of those cells as compared to the ones that were there previously when they had stable vitiligo.
I grew up around uncles that were machinists and arc welders. And the ones that were doing arc welding, I noticed on their arms about the size of a quarter, white blotches on both arms. Not on their hands because they wore gloves, but this is done in the 50s and 60s. Was that caused by that particular manufacturing process?
That's a great question. There's two issues. So with vitiligo, one of my friends gave a good example to this, right? So genetics is how much you look like your family, and environment is how much you look like your neighbor, right? So there's a mixture of both in vitiligo, right? So there is a little bit of genetic predisposition. That's how I explain it to patients, because we know that family inheritance is about 10 or 15%. Identical twins are only 25% cover. Both get vitiligo. So there's clearly an environmental component. And the environmental component is usually some sort of tissue damage. So it's possible that that's the case. The other thing that happens is when you get burns, you basically, that's the other part that's interesting because Resell is also used for that. Because what happens is when you ablate the dermal epidermal junction, you all of a sudden lose these nice pegs and then it's flat. And so in that case, you will see, and you can tell burns look different because they have like dilated pore size and things like that. So part of it might have been burns, part of it might have been vitiligo. I do see dentists, a lot of dentists with vitiligo from the UV light all the time. I see a lot of welders. We see a lot of people that, yeah, so yes.
One question that I had was, is there any connection between like demographics of people with vitiligo and people without? Like you mentioned environmental factors and epigenetics that really come into play when it comes to vitiligo. Then are certain populations of people more predisposed to developing vitiligo or inheriting it?
So there doesn't seem to be, so when we think about skin, we usually think about skin tones and we talk about different types. People who are very fair are like a Fitzpatrick 1. People who are very dark are a Fitzpatrick 6. So it's a continuum like 1 to 6. So when we look across all the skin tones and ethnicities, there doesn't seem to be any predisposition towards vitiligo in one versus the other. But obviously when individuals have darker skin tones, it's more apparent than individuals who have lighter tones. We do, I think just in terms of like talking about demographics, there does seem to be evidence that suggests that individuals who have darker skin tones actually repigment better than individuals with lighter skin tones. And this is due, we think, to kind of like the inherent nature of melanocytes that are in dark-skinned individuals compared to melanocytes in light-skinned individuals.
So what about gender? Because in the really nice video and the picture that you showed, right, that was, I believe, all females except for one individual.
Like 1 or 2 guys, yeah. I don't actually know. I think Dr. Ganesan can mention there's an autoimmunity component to it, and often autoimmune diseases affect women disproportionately compared to men. Anecdotally, in the clinic, we see a lot of men and women. There could be a predisposition towards women because women might actually be more aesthetically concerned for cultural reasons about their appearance. Do you know if there's like a...
I don't think there's a documented change difference. Yeah, so it seems about equal. But I think it affects seeking care as part of it.
Right. Right.
And then just one question about the grafting of skin in order to treat this. One thing that is transplanted is melanocyte stem cells, but I thought that stem cells are limited as you only have a certain amount since birth. So does that mean that grafting can only be done a certain amount of times before it starts to harm other areas?
Yeah, I think they expand, right, when you put them into an area where they aren't. So that's what the tissue control issue is, right? It's like how much they expand. So you can have divisions that are symmetric or divisions that are asymmetric that generate more, and your tissue controls like their division, right? So you might have a situation in normal homeostasis where you have one differentiated cell and one stem cell that maintains and then you stay the same. But in situations where there are none, then they really replicate a lot more and you have more differentiated divisions than maintenance divisions. Yeah.
Which is a great question. But all tissues pretty much have a quiescent stem cell population, right? One that's sitting there and not been activated yet. So when you're replacing, you can drive a lot of activation of what we call a transient amplifier. And if you do that enough, you might deplete the base population, right? That quiescent stem cell population. So it can potentially get to be an issue. Certainly in the central nervous system, in the brain, it's an issue. It made me wonder a question though. So one of the things that tells you whether that's happening is metabolism. And the stem cells that are quiescent tend to be actually glycolytic. And the stem cells that have been activated tend to go towards oxidative phosphorylation, especially as they're wanting to differentiate into what they belong as. But your signature shows activation of a glycolytic pathway, right?
So I guess there's two parts to it, right? So one part is, if we go back, one part is what cells are where, right? Because we have melanocytes and we have keratinocytes. So the keratinocytes, what happens is different. So the keratinocytes, it turns out that these cells that are OXPHOS seem to make the cytokines that suppress the growth of the melanocytes, or don't recruit the melanocytes as effectively than the glycolytic one. And the glycolytic one is a natural thing because the stem cell population in the epidermis is at the base of the epidermis. So that's why you have the depth-dependent change as you go down. Yeah. But it would be great if we could look at melanocytes for the metabolism, but we can't. And the reason is because the melanin is so bright that you can't see anything behind it. Yeah. So you can try to do it with single cell and looking at metabolic signatures, but it's really hard to do in melanocytes. In keratinocytes, it's not hard, but in melanocytes, it is. Yeah.
Are there patients that become stable and they don't repigment versus patients that repigment on their own with time?
Yes. So there are patients that spontaneously repigment on their own with time. Yes. So there are. And then the other part too is that, you know, this whole issue of how far they migrate out. That's why she kind of talked about this whole repigmenting versus non-repigmenting. And that's trying to figure that out, because you have these patients that all of a sudden, well, this spot repigmented, but these didn't. And so that's a great example of, you know, you were asking about controls, a great example of looking at repigmenting versus non-repigmenting versus normal in the same patient. So you can do a three-part comparison. Yeah.
Periodicity of this OXPHOS glycolysis change, does it by any chance correlate with the sort of ridges?
And restate that question in English. Not, that's it, because that, Dr. Guy is a science.
So you have this undulating ridges, right? So the question is that, is the metabolism of the cells in different parts of the epidermis at the ridge different? And that's why. So that's a good question. I don't know if we've directly looked at that. What I can tell you is it does seem like the apical fibroblasts, and this is actually data from Avita, it seems like the apical fibroblasts play a really important role in recapitulating those ridges. So I don't know how much of it's keratinocytes or how much of it's actually the fibroblasts. Because when you graft, that's the one thing why I think this grafting, where you do it with the fibroblasts, makes such a difference than just the melanocytes. Yeah. Yeah.
Stress can cause this traumatic stress, and also the diet, lifestyle, and also supplement can help in mind body medicine. Seems the integrated medicine and functional medicine can help with this.
Yeah, with vitiligo, as Dr. Ganesan mentioned earlier, there's a genetic component to it, right? You have some kind of predisposition. I shouldn't be doing this. You have some kind of predisposition towards it genetically, but there has to be something that kind of, you've got your scales, and there's got to be something that, the factor that tips the scales. And it can be emotional stress. In clinic, we've had people, for example, there was like a young woman who went through a lot of emotional duress and then developed vitiligo afterwards. For some people, it's a physical stressor, like they actually have a damage like to the exterior of their skin, or it's another kind of physical stressor in that they've had a really significant illness, like some kind of systemic or other illness, and then that's manifesting as vitiligo as well.
And there are vitamins that seem to help with stabilizing the disease. It's hard to measure exactly, but you know the way we measure it is if you put somebody on treatment and when they're on certain vitamin regimens, they actually improve faster than those who aren't. So, and that's most likely has to do with what's needed to regenerate. So there is some evidence for In fact, I give patients a list of vitamins when I go home.
This one is loud, wow.
Yeah.
So I think we'll probably call it there. I would like to make just one more pitch though, because it is a part of our community outreach here, to make a distinction that's really about academic medical centers, right? And so I hope one of the things that you come away with tonight is the idea that all of these questions that you guys have started to answer come out of being in a situation where you can ask them, right? And if you're working at a private dermatology clinic in Newport Beach, not that that is not valuable work, but your interactions with patients in this kind of clinical population may not be the same, and your ability to come back to the lab and start pulling apart the threads that tell us the biology that informs the next steps for how can you move forward to clinical translation, right? And think about helping folks who are in this situation is vastly changed, right? And so this is the power of being at a university. And as you all know, we're really big on universities here. So with that, please thank our, yeah.
With regards to that. The other part too that I would say is even though you work really hard, there's a lot of job satisfaction with this too. Because the patients who come in to see you, they need you. It's not like they can go see anybody else. When they come to see us, they're there for a reason. And that relationship is very different than in private practice or anywhere else. So that's something that we don't think about, but it's very true.
Yeah, perfect. Thank you so much. So please help me thank our speakers for this evening, and thank you all very much for attending. Happy holidays.